Screencast, where this time we'll be discussing population genetics and the Hardy-Weinberg equilibrium, Hardy-Weinberg theorem. And so by the end of this, you should be able to describe and explain what we mean by a gene or an allele pool, what we mean by allele frequency and how to calculate that, be able to list and describe the 5 conditions that are necessary for a population to be in Hardy-Weinberg equilibrium, and then to evaluate and apply evidence to quantitatively investigate the role of natural selection in evolution. So we're going to try to quantify evolution here of a population, and then evaluate and apply mathematical routines to determine the evolution of a population. And again, this mathematical routine here is going to be involving calculating a Hardy-Weinberg equilibrium, calculating Hardy-Weinberg allele frequencies, etc. here. So let's get started. And single genes with discrete alleles determine some phenotypes of organisms in populations. So for example, Mendel was looking at seed color, and there was one gene and there were 2 alleles here, either a yellow one or a green one, and you had either or situations here. Thomas Hunt Morgan saw the same, a lot of similar phenotypes here with flies. You had wild type, body type, and an ebony, so you had either or here. You didn't really have any mixing. And so let's look at a hypothetical population here of flowers and flower color, where we have red flowers and we have white flowers. Right? And we'll just look at alleles here, and we'll say that R is dominant to red is dominant to white, and we use the big R and little r here. And of course, noting that we could have used a different notation as well, just being R and W, right? But we're going to use the big R and the little r here. So this is an image of sort of what we mean by an allele pool. So the allele pool or the gene pool, you'll often hear gene pool, but more specifically I like to think of it as an allele pool, is all of the alleles present in a population. And a population is defined biologically as a group of the same, same species, right, in the same area at the same time, right? And so this is what we mean by a population. And again, the same species, meaning they can interbreed. You can interbreed and form viable, that is living, and fertile offspring. So this is kind of the biological species definition. This is the test. Can you mate 2 organisms together, and can you interbreed them, that is, and form viable, that is living, and fertile offspring? Right? So if we look at the allele pool, here we see, let's define this as the borders of the population. We'll get these guys into here. And so this is the same area. So all of the alleles that are in an area at any one given time. And you see that we have big R alleles and we have little r alleles. So we have 2 different types of alleles here. So all the alleles present is the allele pool or the gene pool. And we're going to say down here that each big R here represents 5. Each big R, each little r represents 5 alleles there. Now, this is a little awkward to think about, but alleles in the populations are stored on chromosomes that of the individuals, right? So we're going to assume that these individuals are diploid for the sake of argument here. So really, an organism is a storehouse of alleles. That's one kind of crude way to think about it, but each individual here, each red individual, is either going to be homozygous for red, is going to be heterozygous, or would be homozygous for white. And if you add up all of these alleles in this population, right, that are here, you see that they're going to be the same number as there are here. Now, each individual has space for 2 alleles because, of course, they are diploid. So one allele is going to be on one chromosome, one homolog, and the other allele is on the other homolog, right? In this case, they're going to have 2 big R's. In the case of this heterozygote over here, one is going to be big and one's going to be little. And in the case of these homozygotes here, you have centromere allele or locus, right? And you're going to have 2 little r's there. So again, each one of these plants now is, each red one or each white one represents 5 individuals. So if we do the math here, the frequencies of these alleles can be calculated, right? And so the allele frequencies are calculated, we can calculate a phenotype frequency, we can calculate genotype frequency, we can calculate allele frequencies, and it's ultimately allele frequencies that we want to know. But in any environment, we only really see phenotypes. We can't look at an individual and know whether it's what his genotype is, unless, of course, it's the recessive phenotype. Now, this Hardy-Weinberg equilibrium, this Hardy-Weinberg theorem describes a non-evolving population by comparing allele frequencies, right, over the generations. So we need to get down to this allele frequency bit here. So how do we figure out the allele frequency? Well, again, here is what we see. We see phenotypes. We can infer genotypes from some of these, right? And if we go back and count up, we had 500 total plants there. So if you go and you look at this, you should be able, if you wanted to, you could go and calculate that there are 500 total plants there. And of those 500 total plants, 20 of them are going to be white, and then the balance of them are going to be red. Now, we don't know, the red ones, we don't know if they're going to be homozygous or heterozygous there just by looking at them. But for the sake of argument, let's say that we counted them up here for the moment, and we know that there are 320 of these dominants, homozygous dominants, and then 160 of the heterozygotes. Well, to calculate the genotype frequency, we take the total number of organisms in the population and divide that into the number that are the genotype that we want, right? But what we're really after down here is the allele frequencies. This is what we want to know, because that's what Hardy-Weinberg is going to be comparing, the allele frequencies, right? So we define the frequency of one of the alleles as P, right? And this would be the frequency of the dominant allele, and the frequency of the recessive allele we're going to define as Q, right here. Now, how do we go and calculate this? Well, why do we know that there are, again, the frequency is going to be, to determine this, is the number of the alleles, the allele type that we have, over the total number of alleles in the population. So here we see that there are 1,000 alleles. Well, how do we get that 1,000 alleles? Because we know from the example here, they're giving us that there are 500 individuals, and each of those individuals is diploid, right? And so what we know then is that if those 500 individuals, if 320 of those 500 individuals are big R, that represents 640 big R's. Right? We time this by 2 because they are diploid. And these heterozygotes here, there's 160 heterozygotes, so 160 of these alleles in these diploid organisms are also big R. So we go 640 plus 160 gets us to this 800. Right? And then 800 divided by 1,000 is, of course, 0.8. So we say that P here is equal to 0.8. Right? Now, the remaining alleles, 160 here from this heterozygote and 40 here from these homozygous recessives, we know that there are 20 of them were white, and so those 20 individuals each have two alleles, and they are little r's. And so, again, this is because the organisms are diploid. Right? And so we get 40 of these plus 160 gets us to 200 out of 1,000, and that's 0.2. Now, what you can do here, of course, is that P plus Q is equal to 1 all the time. Right? So this is talking about allele frequencies. This is one equation that we can use. Now you'll notice up here that this big R is really P times P, which is really P squared. This big R little r is P times R, and there's more than one way to get that, right? because you can also do this, right? And so this is there's two of those, and then R times R is really R squared. So if you're looking at genotype frequencies, you know that P squared plus 2PQ plus Q squared is also equal to 1, right? And so this is if you're given genotypes, and if you are given alleles, right? And then phenotype frequencies, which you don't often see discussed, right? But the phenotype frequencies, the dominant would be P squared plus 2PQ, and then the recessive is just going to be Q squared. Now, more on this later, and we'll deal with these in class and we'll show you how to convert back and forth between them. But you definitely always want to try to get to here, right, and figure out what P is and what Q is equal to. Right? Now, this is the gene pool of the parent population. Now, let's say that these parents here, these individuals, will mate with one another, right? And the likelihood of getting a big R, right, here is 0.8, because the allele frequency, this is of the total alleles, 8/10 of them are big R, and then 2/10 of them are little r. Right? And so the likelihood of getting this combination in the next generation is 0.8 times 0.8, or 0.64, so P squared is equal to 0.64. The likelihood of getting, probability of getting a heterozygote is 0.16 plus 0.16, or 0.32, and then the probability of getting a recessive phenotype is 0.04 here. And so getting the genotype frequencies of the next generation, this is what we're doing here. We're predicting what we should get, in the next generation. Here, we can, if we know P and Q of the parent generation, we should then predict what's going to happen, what we would see in the next generation. And if you do all this out, right, you figure out here that Q squared is 0.04, right? So Q squared is 0.04. If you take the square root of this, and you end up getting Q is equal to 0.2. Right? So this is where kids sometimes make mistakes, is they don't know how to do this, that you actually, 0.2 squared is 0.04. A mistake that kids make is that 0.2 squared, and they say is 0.4, but this, of course, is not correct. So this is an error to watch out for. All right. So if Q is 0.2, then P is 0.8, and we say then that in the next generation, the P and the Q did not change. Right? They stayed the same as the parent generation there. And so this population then is described as being in Hardy-Weinberg equilibrium, meaning that the P and the Q did not change. The frequencies of the alleles over the generations did not change in that population. Right? So some phenotypes that are determined by multiple genes, each with multiple alleles, but still interbreeding of organisms passes on those alleles to the next generation. Right? And so what we're seeing is that not a discrete situation here, but rather a variation here showing a normal distribution, right? Where you get shades here of, say, pigmentation or in height. Right? Multiple alleles here are determining this, and so it's not as simple sometimes. Right? However, you can use some of the same principles here to predict what should happen in the next generation. An extreme example here would be using cystic fibrosis. So there are 4 alleles in the population of humans that exist for cystic fibrosis. Now, just real quick on cystic fibrosis, this is a water and ion channel. Water and chloride ion channel, and in normal individuals, this ion channel here is open, and water and chloride ion are free to move across here, but in individuals that inherit a certain combination of alleles, the channel is altered right here, and you end up producing this thick mucus that makes it difficult to breathe. So these ion channels are in the trachea and the bronchioles of the lungs. Trachea, bronchi, bronchioles of the lungs here, and it makes it difficult for individuals with cystic fibrosis to breathe. But if we look at the different alleles that are present here, CFTR. CFTR, by the way, is the gene that is this is the CFTR protein here channel. So these are the genes, the alleles, one gene but multiple alleles that exist in the population. So we've got CFTR1 through CFTR4, and again, this is the allele pool. This is kind of what it would look like. But those alleles are on chromosomes of individuals, and again, we're going to assume diploid. So here I'm just showing you that on chromosome number 7, this is the locus, right? And diploid individuals inherit 2 of those alleles, right? And so this would be a homozygote for 1, this would be a homozygote, this would be a 1,4 heterozygote, this would be a 2,1 heterozygote, here's a 2,4 heterozygote, here's a 3,3 homozygote, etc. Right? So these individuals exist in the population. Each one possesses 2 alleles, right? And so we can calculate genotype frequencies, we can calculate allele frequencies, we can calculate phenotype frequencies from these. Right? Let's go right to the allele frequencies, and if you take all those from the previous image, right, we got 24 CFTR1s, 32 CFTR2s, 4 and 8 for the CFTR3 and CFTR4 respectively. So if you understood the previous slides here, that means that we have 68 total alleles in the population because there are 34 diploid individuals. So now we're not just going to have P and Q, we have P, Q, R, and S here, where P is the frequency of CFTR1 and CFTR2, CFTR3, CFTR4 respectively. So can you calculate up here what is the frequency of CFTR1, what will be the frequency of CFTR2, 3, and 4 respectively, and you can show your work down here. We can do that in class as well. Okay, so there's something to practice with. And now finally, if the population is going to be in Hardy-Weinberg equilibrium, we say that the allele frequencies are not changing, and then five situations must be true, must be met in order for this to happen. And so you should be able to identify these five situations here. First of all, that there should be no natural selection. No natural selection. There should be random mating between the individuals. Again, if we don't want the allele frequencies to change, there should be random mating. We should have sufficiently large population size, and this will limit what is known as genetic drift. Limit genetic drift. They often describe this, the next one, as no immigration. Right? No immigration or emigration. That is, no individuals coming into or leaving the population. They sometimes refer to this as no migration, but I like to think of it as immigration or emigration. Migration means it implies to me anyway that the whole population is moving from one area to the next, like a breeding ground to a feeding ground, etc. But the immigration-emigration bit here is to limit. You don't want any what is known as gene flow, right? So there's no gene flow here. That is alleles entering or leaving the population. And finally here, no mutation. Right? No mutations to those alleles should exist, because if you mutate the allele, then you change the allele, and then that is going to, of course, change the frequency of them. So these 5 conditions must be met in order for a population to be in Hardy-Weinberg equilibrium. That is, no changing of allele frequencies over the generations. Right? The only one, of course, which will sort of adapt a population to its environment is natural selection, and we'll talk a little bit more about that, of course, in class. All right? So, here's a population on the left. Here's the parent generation right here. So take your time here and practice again, calculating the P and the Q for this generation. And then what's happening here is that the individuals in white, only some of these are surviving and reproducing. And so here's the F1 generation here. And you know the phenotypes, you know the genotypes. So again, you can calculate P and the Q there. And then again, we have only some individuals reproducing here. And so this F2 generation, calculate the P and the Q here. And the question is this population evolving? And if it's evolving, how do you come to that conclusion? What evidence do you have to support or justify that? And if it's not evolving, what evidence or justification do you have to support that? Okay. So some things to practice there with the CFTR, calculating the allele frequencies, and then here in this example, calculating the allele frequencies and then coming to a conclusion about that. Okay, so to sum things up here, you should be able to now describe what is meant by a gene or an allele pool, know how to describe and calculate the allele frequency, and the 5 conditions that must be met in order for a population to be in Hardy-Weinberg equilibrium that is not evolving. And then evaluate and apply evidence to quantitatively investigate the role of natural selection and evolution, and mathematical routines, and that again is calculating the P and the Q, those Hardy-Weinberg equilibrium situations there, and to know and determine whether or not a population is evolving. Okay, the rest of this, we're going to do this in class. Here's the example that we're going to work through in class, right, with tasters and non-tasters. Okay, so thanks for listening, and look at that pre-lab and bring your questions to class. Thanks.